organic phosphate, we've got a ribose sugar, and we've got a nitrogenous adenine base, folks. And so that's what ATP is made of. Moving on, this is the spec. I'm not going to read it out. Instead, I'm going to talk to you a little bit about that shows. Before that, this slide here is a bit maybe beyond what we need to know, but I think it's really helpful. Adenine and ribose together is where we get the name adenosine. So the name of the whole thing, ATP, is adenosine triphosphate. Well, you say, hold on, why is it not adenine? Well, it's just an abbreviation, adenine ribose triphosphate. It's a bit quicker to say adenosine triphosphate. If I only had one phosphate on, we could call that a nucleotide. We could also call that adenosine monophosphate. We could call it, with two on, adenosine diphosphate, and if there were three on it, we could call it adenosine triphosphate. So it could be called ATP, ADP, or AMP. You're not going to call us AMP too much. Okay, so that's just a bit of background for you. So see, what you will come across is ATP and ADP, a heck of a lot. So this is still on a bit of a recap of year 12 at this point. So let's have a look, folks. We have ATP here, okay? Don't need to worry about those bonds. And when we convert to ADP here, we are going to get one phosphate group, and we're going to get some energy. And this is a hydrolysis reaction, okay? So we're going to require water for this, okay? In the other, and energy. Sorry, no, we're going to get energy, sorry. In the other direction, it's going to require energy to reform ATP and an enzyme. So it's going to require one type of enzyme to do this reaction, different type of enzyme. So what was this enzyme? This was ATP synthase. Anyone remember the name of this enzyme here?
ATP hydrolase.
That's the one, okay? And so very similar to everything else in your biochemistry unit in year 12, hydrolysis, breaking down with water. So we're breaking down these bonds with water and an enzyme, and we're resynthesizing these bonds. We don't need water on that one, but we do need energy in this case, okay? And so what is ATP used for? So reproduction, cell division. This looks like reproduction, but this is actually replication, as in making new molecules, what we're going to call synthesis of molecule. What we're synthesizing there, a new strand of DNA. So synthesis requires energy, as I've just said. For example, ADP to ATP requires energy, okay? Forming a new strand of DNA requires energy. Making a protein requires energy, etc. Synthesis. And then this is supposed to be showing, it's like looking in the mirror, isn't it? This is supposed to be showing movement, okay? That's not growth. These are muscle fibers, they're going to move. Anywhere you see movement in nature, flagella, muscles, anything, it's ATP for movement. So movement is a good one. And then, as Liam said, active transport, folks, requires energy. So those are the main categories. All right. Synthesis, muscle contraction, cell division, light independent reactions of photosynthesis, first stage of glycolysis, which we're going to do today, and active transport. Right, well, this is what George has just described that you're well familiar with, and I've written in brackets. This equation is a simplification of a process with 30 separate steps, folks. So it's not that your GCSE was wrong. It's a very, very simplified version. This energy could be lost as heat or trapped and stored as ATP, okay? In terms of this, we've already talked about this. So the only thing that might puzzle you about this process, look, I've written it at the bottom. So we've got glucose and we've turned it into ATP. And so physics-wise, we've turned chemical energy in glucose to chemical energy in ATP. So we haven't really achieved anything new there, okay? We've taken the same form of energy and transferred it into the same form of energy. So my analogy is going to get a bit weak here, but so my analogy was going to be, let's imagine glucose is a 20-pound note and ATP is a £1 coin, okay? Now the only problem with that analogy is you've all gone contactless these days, so I don't know, let's imagine glucose is a 20-pound note and ATP is like a contactless card. You just whack little bits of energy out as needed, okay? So there's more to it than that, but that's just an analogy there for you, okay? So the energy is stored in the bonds, okay? That's a very familiar use. So as well as being small amounts of energy, as I've just said, been broken down, it's also manageable and transportable. And so here are a few things that you would perhaps remember. So it's much smaller amounts of energy than glucose. It's much more easily broken down than glucose. We can pass it in and out of the cell, whereas glucose can only get into the cell via active transport. Small and soluble, easily transported around the body, and we transfer energy with these phosphate groups. Okay, so that's a good source of energy. Here's another recap. These folds, cristae, in the inner membrane of the mitochondria have a very large surface area to maximize respiration. And as I say, not today, but we'll come on to it, that there's a whole load of reactions going on this inner membrane, okay? There are these embedded proteins in this inner membrane that are carrying out the last stage of respiration, okay? And so having this super high surface area means more respiration, folks. Okay, so that's the spec on this. I do need to talk a little bit longer, sorry about this. This is a reasonably tricky, biochemistry is a reasonably tricky topic in terms of there's a lot to remember. So if I can get anything decent out of this, I'll put it up on my YouTube channel that some of you will have seen. It's not very good, but I'll put it up there because you may need to go over it more than once. So there are 3 coenzymes. So what is a coenzyme? It's a molecule that aids an enzyme. So in this case, the enzyme is going to catalyze the reaction, and then the product of the reaction is going to be taken away by the coenzyme. And the ones that you're going to use a lot, and especially today, we're going to be mainly talking about NAD. Okay, you would have come across that in photosynthesis as well. So NAD, coenzyme A, and FAD transfer hydrogen from one molecule to another, so it can oxidize or reduce a molecule, whereas the coenzyme A is going to transfer acetate. Okay, so the enzyme is going to do the reaction, and then the coenzyme is going to take the hydrogen. The hydrogen is not going to just float off into the cytoplasm. It's not going to stay on the enzyme. It's going to be released from the enzyme onto the coenzyme.
The NAD in this, the coenzyme in respiration is NAD, but in photosynthesis it's NAD PH, when it's been reduced. So don't get them confused, because they are, if you wrote about NADP in respiration, it'd be incorrect. It's just NAD. It doesn't have the phosphate attached.
Yeah, that's a good point. There's all kinds of marks got lost every year with NADP and NADH. It's
a horrible topic in terms of.
Okay, so first of all, a bit of chemistry. Okay, so we need to bear this in mind. when something is reduced, it may have gained electrons, gained hydrogen, lost oxygen. When something is oxidized, it may have lost electrons, lost hydrogen, gained oxygen. Gained oxygen was the original meaning of oxidation when they started burning stuff. And they said, oh, I've burnt carbon, it's become carbon dioxide, it's oxidized. And they thought of a new word. Then we understood it to mean loss of electrons, as you will in chemistry all the time. In this topic, you need to remember oil rig. Oxidation is loss of electrons and also H+. Loss of hydrogen is oxidation. So that's something that you pure chemists may forget, because you don't come across that so much in chemistry, that loss and gain of hydrogen, we can include that in oxidation. All right, so we have to remember that. And so just oil rig, folks, works for electrons and works for hydrogen. I'm going to give you a big overview of the whole respiration topic. So we start with glucose here, and on the back here you can see we've got the various parts of the cell. So glucose actively transported into the cell. It's a 6-carbon species, and all of this 1, 2, 3, 4 is called glycolysis, and that's what we're going to be talking about today. And it will end up with pyruvate, which is a 3-carbon molecule. Now this 3-carbon molecule is going to go through the mitochondrial membranes. There's 2 of them, okay, inner and outer. Okay, and form acetyl coenzyme, and this is going to be called the link reaction, folks. And so that's happening in the matrix. And then there's something called the Krebs cycle here. That's going to be called the matrix. That's going to occur in the matrix. And then we're going to have the last part here is the respiratory chain there. And that's happening, as I mentioned earlier, in the proteins on the cell membrane, and that's why we've got all these crista. And so all of this process of many reactions is just there to make ATP. Okay, so we're not even talking about, we're not even going to talk about, you know, how the ATP is used. It's just how glucose is used to make ATP. That's what these series of lessons is about. Okay, so that's a very short kind of overview of the process. From what I've just said, where is this occurring in the cell? The other thing, you can keep an eye on the reactions here. We've got glucose C6H12O6 going to pyruvate C3H4O3. Okay, where does that occur? That's in the cytoplasm, folks, of the cell, not the mitochondria. Krebs cycle will occur in the matrix, and you'll see we've started to get CO2 given off. And then respiratory train in the membrane there. I've missed out link reaction, but that's also in the matrix, folks. You'll see this is where the oxygen starts getting involved, okay? So we actually have CO2 being produced before oxygen is ever used up, folks. So actually the production of CO2 is independent of the using up of oxygen, okay? And so the final product there is water and lots of ATP, okay? As I say, 30 reactions to get there, and we're just going to study a few of them. All right. So looking at glycolysis, we've got 2 stages. We've got phosphorylation, which is adding a phosphate group, and then oxidation, which is going to be the loss of hydrogen. Okay. So if you're a non-chemist, don't worry too much about these next door diagrams. I just find them a lot easier. So in glycolysis, we're going to start off with glucose. we're going to phosphorylate it here to form glucose phosphate. And we're going to phosphorylate it again to form glucose bisphosphate. Okay. And so our first point about this will be that's a bad start if you're trying to make ATP, because as you're going to see, we take glucose. Where are we going to get these two phosphates from? We're going to convert ATP to ADP. We're off to a bad start because we're supposed to be making ATP, not using it up. But like many things in life, you've got to put something in to get something out, haven't you? You've got to spend your money to make money. So we're going to spend a bit of money here by putting phosphate on it. All right. And so those of you at chemists will understand that what we're doing here, we're trying to split the glucose is too big. Let's be honest about it. It's packed with energy, it's too big. We're trying to split it into these 3 carbon chain molecules, folks. And so, you know, we haven't got a Bunsen burner to split it in half and some nasty heavy metal chemicals. So what's another way to split something? Well, look at all these charges going on, folks. PO3 2-. Look at all these oxygens that are bound to have an influence. And so those of you who are more into your chemistry will see, well, there's going to be a cut. See, there's going to be a cut there, and there needs to be a cut in the bond there. Okay. So as was said in photosynthesis, this adding of the phosphate is going to help with that breaking of the glucose down into these smaller molecules. So we don't need to go into that further than that, but again, that is just an overview of it for the chemists, and we need to look at more in detail now. So let's look at it in detail. So glucose is phosphorylated using a phosphate from ATP. There it is. Okay, and so we get a 6-carbon glucose turn into glucose phosphate, and we use up an ATP, and then we do so and use up another ATP, and we get hexose bisphosphate. The bi or bisphosphate, bisphosphate. Bi meaning 2, or 2 phosphates on it there. Then the molecule is going to split into 2 triose phosphates. Okay, so what we need to remember here is we've 1 molecule of glucose has made 1 molecule of hexose bisphosphate, and we've used up 2 ATP molecules. Okay, but the important point here is triose phosphate is oxidized. Okay, it's going to lose hydrogen, forming the 2 molecules of pyruvate and getting back some of the ATP that's expanded, and then we have NAD will go to reduced NAD. So I've also called NADH there. Okay. That 4 ATP is a bit weird. I'd like to swerve that. I'll come back to that. You'll be thinking, well, why is that not 2 ATP? Well, because you had hexose bisphosphate going to 2 triose phosphate, so you'd assume there's 1 phosphate on each. So you'd assume you were going to get 2 ATP there. I don't really want to deal with that at the moment. It goes into a series of other reactions, okay? But you have to learn that 4 ATP is produced there when you might expect 2 ATP to be produced there. So NAD being the coenzyme is going to collect the hydrogen ions from the oxidized triose phosphate. We've got 2 reduced NAD, we've got 4 ATP, 2 we used up here, so it's a net gain of 2 ATP. So a bit of a recap here. So what have we got out? We started with glucose. What do we end up with? We ended up with 2 reduced NAD. We ended up with 2 pyruvate. Why do we end up with 2? Because we started off with a 6-carbon molecule. We ended up with 2 3-carbon molecules. We ended up with a net gain of 2 ATP. We 4 ATP. ATP was put in at the start. Okay, where did the NAD go? It was used for oxidative phosphorylation. Where the pyruvate go? This is now going to go into the link reaction. So what that tells you is the pyruvate now can cross the cell membranes of the mitochondria, okay, by active transport.